everyone. My name is Xinyue Chen. I'm currently doing a postdoc in the University of Sheffield with Professor Jamie Hobbs. Thanks for offering this chance to talk about our research. We've listened to many fantastic talks of revealing very fine details by AFM these days. Differently, our study used AFM on giant complex tissues. So mechanics has been widely agreed to play a vital role in cancer, but there is a stiffness paradox. Single culture cancer cells have repeatedly been shown to be more compliant than their healthy counterpart, while tumors being stiffer than the corresponding healthy tissues. However, in 2012, a Swiss group measured the stiffness on fresh patient biopsy using AFM, reported that the core cancerous lesion is actually softer than surrounding healthy tissues. It's a great example showing what kind of samples and how you prepare the samples strongly affect the mechanical characterization. To understand cell behaviors in nature, it's very useful to measure in the context of near intact complex tissues, as we are focusing. So we study the breast cancer, which is the most common one in the UK. Cancer cells can depart from the primary site, circulate, and home to a distant organ to develop metastatic tumor, which is incurable to date. Bone is involved in about 70% of all breast cancer metastases. We aim for understanding the role of mechanics in such late-stage cancer in bone. It is almost impossible to work on human biopsy in this case, so the mouse model was used instead. How to access the tumor in complex bone structure while maintaining the intactness. The simpler, the better. We use this custom design tool with razor blade to split the bone by simply chopping. The bone will be immobilized while dental glue and measured under nearly physiological conditions. Here is an image of one healthy bone sample in rail. The outline area called bone metaphysis is where most of the metastatic breast cancer cells favor to home. Experimental bone metastatic tumor were established by cardiac injecting MDA-MB231 cells, an invasive breast cancer cell line, to the mice. The occurrence of the metastatic outgrowth was preliminarily determined by in vivo bioluminescence imaging. Once the signals observed behind this were detected, and the metastatic tumor could be targeted with the aid of customized in situ top view microscopy while GFP fluorescence. AFM measurements were applied on top of the metastatic tumor and compared to either other cancer models or tissue environment. Our mechanical measurements started from the most commonly used method, fitting the Hertz-Natter model to force indentation curves to get the Young's modulus. A spherical colloidal probe was used. The diameter was about 25 micrometer, which is giant. This is not only improving the fault sensitivity, but also more biologically relevant because the size is comparable to a cell. Such fitting is not perfect, regardless of correcting the error in contact point determination. This is reasonable considering most of biological tissues are viscoelastic instead of pure elastic. To take the viscous effect into account, we also applied creep measurement, where track the deformation change under constant force. The fitting equation was derived based on Kelvin-Vogt model, properly consider the 3D instead of 1D deformation. We've also tried a 3-element standard linear solid model, but the Kelvin-Vogt seems being sufficient from our results. We have done both types of poking at selected positions within regions of interest and summarize these mechanical distributions. Here are the Young's modulus from Hertz-Natter model, the Young's modulus and viscosity from Kelvin-Vogt model of the metastatic tumor, non-orthotopic tumor that is formed around the subcutaneous injection site, and the cultured cancer cells on Petri dish. Please note these are log scales, so the moduli of tumors cover more than 2 orders of magnitude, which means they are mechanically heterogeneous. box plots, and histograms are probably easier to see the comparison. We can see metastatic tumor is overall extremely confined, with moduli at only a few tens Pascal. Mechanical properties of cultured cell model are over an order of magnitude higher than those of tumors, indicating the importance of a proper 3D environment and acellular components in cancer mechanics. The metastatic tumor is considerably more confined than the subcutaneous tumor. This can be explained by either metastasis requires a stiffness selection or the bone niches applied extra mechanical alterations to hold cancer cells. Next slide shows the mechanical properties of the metastatic tumor, its surrounding bone metaphyses, and metaphyses from healthy bones without cancer injection. Remember the log scales, and we can see the mechanical heterogeneity as well. The metastatic tumor was found to be significantly more confined than its surrounding environment, even though the bone metaphysis is also very confined. Also, no statistically significant differences were found from the metaphysis with or without tumor, indicating the metastatic tumor did not mechanically alter the environment at a mesoscale distance. Therefore, we conclude the mechanical events between tumors and the metastatic size, if there is any, would occur within a very short scale at their interfaces. In future studies, it will be useful to characterize the structure mechanics correlation across the tumor microenvironment interface. We haven't got results at such interface yet, but I can quickly introduce some general ideas of what we can and will do. So on one hand, we can successfully map the local mechanical architecture with subcellular resolution on fresh complex tissues. This is an example on a healthy bone. You can see the
You have 3 minutes left.
Yes, at such supercellular scales has already covered orders of magnitude. This unique architecture may provide, for example, soft channels for cell motions. We will do the same at the tumor interface. On the other hand, combining optical microscopy is necessary for identifying the measured tissue components. Currently, we can roughly correlate with high-resolution post-FM confocal images using the cantilever position and special bone patterns obtained from our in-situ top-view microscope images. And we are aiming to further improve our in-situ setup later. Okay, some take-home messages. First, by different models, we know in vitro models we may not well present tumor in nature. Then the metastatic tumor to bone is still significantly softer than its surrounding tissue environment. Last but not least, the metastatic tumor does not largely alter the mechanical properties of its surrounding tissues. These findings, we thought they are inspiring for designing more rationalized cancer research models or mechanical interventions as anti-cancer drugs or treatments. For future developments, we believe it is very important to seek a way of establishing in-situ top-view optical combination with significantly improved spatial and temporal resolutions. Only with this, we will be able to know not only what we are measuring, but also how, by monitoring the displacement of the different tissue components. So that's all I plan to present today. I would like to thank all colleagues in Sheffield working on this project, as well as other colleagues in the HOPS group. The funding was from CR UK and EPSRC. Thanks for your attention, and I'm more than happy to take any questions later on discussion table.